Hi everyone, this is Courtney Wallner and I'm going to present to you my bioinformatics project for AGRO 215. The focus of my project was the blotched coat pattern on domestic tabby cats caused by a mutation in the TACPEP gene. So the domestic cat, or Felis catus, has many different breeds and different markings similar to dogs. But some cat breeders are noticing that after mating two purebred mackerel-coated tabby cats, which is the striped kitties that we see in these pictures, some of their kittens were coming out blotchy, like the kittens in these pictures down here. This encouraged breeders to work with scientists and geneticists to figure out what exactly was happening, and more importantly, if these blotchy cats indicated something fatal. But what they found out was that these blotched cats had mutation in the TACPEP, or the transmembrane aminopeptidase Q gene. This gene will also be referred to as the LVRN gene throughout this presentation, which is in the same family, but it's a little bit less specific. However, it has more information available about it. However, the TACPEP nor the LVRN gene encode the protein that affects the coat color, but rather it is the first step in a two-step catabolic pathway. The mutation in the TACPEP gene affects the expression of the endothelin-3 gene, abbreviated as the EDN3 gene, which coordinates the localized color differences. It was found that the blotchy pattern behaved in an autosomal recessive type way, meaning that heterozygous mackerel-coated parents have a 25% chance of having blotchy kittens. This mutation is actually not harmful in any way to the cat, and in fact, the TACPEP gene belongs to the gene family involved in immunity. The aminopeptidase that the gene encodes is a receptor, and the mutation actually inhibits some viruses from recognizing the receptor and gaining entry to the cell. It is thought that this mutation is stuck around due to the increased immunity that homozygote recessive and heterozygote cats have. Unfortunately, although it's a blessing to the kittens, it might be a curse to the kitty's wild cousin, the cheetah. The blotching has been found in cheetahs as well, and until genetic testing was done on the tabby cats, it was found that the blotchy cheetah was actually an entirely separate species known as the king's cheetah. After some testing, though, it was confirmed that the reason for this blotching was due to a mutation in the TACPEP gene, just like in the cats. It does look really cool, however, the cheetahs, being wild predators, rely on camouflage in order not to be spotted by their prey. This mutation may aid in preventing infection, but it actually may hinder the big cat's ability to hunt. So, why does it matter? Although this mutation isn't lethal by any means, many people are interested in preserving this relatively and naturally occurring trait. In the world of breeding, diversity has been handpicked by humans for thousands of years, so any primal traits have mostly been brought out. The presence of this mutation in cheetahs informs us that this gene and mutation stretches way back in time and connects across many different species. Therefore, it has value that no amount of artificial selection can contain. The mutation in this gene may also inform us of any eventual discoverable repercussions, as the LVRN gene spans across many different mammals, including dogs, pigs, and humans too. If a problem can be identified in a cat, it can be solved in any case any humans may suffer due to a mutation, which would most likely affect the pigments, such as like in the disease vitiligo. And of course, this mutation is important because it is a way of conserving genetic diversity within the population. Inbreeding has essentially wiped out any genetic diversity in cheetahs due to a bottleneck effect that occurred around 10,000 years ago in the last ice age. There were so few cheetahs left afterwards that the entire population alive nowadays is more than 99% genetically identical. That's like every single cheetah being an identical twin to each other. The TACPEP mutation, however, though seemingly unimportant, is actually a great indication that diversity does exist among cheetahs and can give them a fighting chance against extinction. And on a more personal level, I plan on becoming a veterinarian someday, so I'll be seeing a lot of cats. I can make informed breeding decisions based on that knowledge of this mutation and could maybe identify similar mutations if they arise in any other of my patients. Also, I love cats. This is my cat Mario, looking adorable, and I figured I would include her in this presentation. So, my bioinformatics project starts here at PubMed Central. It is a scientific database that is part of the National Center for Biotechnology Information, or the NCBI. I first became interested in this gene after reading someone comment on a cute picture of a blotched tabby cat. The commenter mentioned that this was caused by a mutation in the TACPEP gene. Obviously, a stranger's word on the internet isn't worth much, so I decided to find some scientific backing to his claim. I searched the word TACPEP in the search bar, and only 2 of thousands of articles available on this site came up. I lucked out, and the first result was exactly what I was looking for. The article, titled Specifying and Sustaining Pigment in Domestic and Wild Cats, detailed how this coat pattern exemplifies diversity amongst individuals and unity amongst species, and then goes on to explain the two-step method of expression of this odd phenotype. If you're interested in reading it yourself, the PMID is 22997338. So, now that I confirmed the name of this gene, my next step took me to UniProt, or the Universal Protein Resource Bank. It is a database that allows you to search for your gene of interest, determine its nucleotide sequence and therefore amino acid sequence, and then compare it across species. It gives me information such as the protein name, function, and other potentially useful information. Like I said earlier, a more common and less specific name for this TACPEP gene is the LVRN gene, which is what I had to search for on UniProt in order to find my protein in my species of interest, the cats. The next thing I did was run a FASTA search and then a BLAST search. So this is what the search results are going to look like if you do look up this on the UniProt website. This is what a FASTA looks like, and then this is what your BLAST results look like, and I'm going to explain that a little bit more. So here is my FASTA results for the gene, which gives me the amino acid sequence of the protein it encodes. FASTA is a local alignment tool used for comparing sequences across different organisms. The first 6 letters of my sequence, which would be YYGLKR, actually encodes for the following amino acids: tyrosine, then tyrosine, glycine, leucine, lysine, and arginine. If I had the time and interest, I could actually decode the entire thing. So this is extremely useful information depending on the context a researcher or geneticist may have. So the next thing I did was a BLAST search. The BLAST, or the Basic Local Alignment Search Tool, will compare biological sequences, whether it's an amino acid sequence, DNA nucleotide sequence, or anything else like that. Lucky for me, there was a direct link from my UniProt search for the Felis catus LVRN gene, so I clicked and here are the results. These results are telling me how similar other sequences are in other organisms. The cat obviously has a 100% sequence match, go figure. The next organism with an 85% similarity with the gene is the domesticated dog. Following that is a ferret, then 2 subspecies of giant panda, and a pig. We can see that the results get more and more dissimilar, and the organism becomes less and less cat-like. Interestingly, though, the first 3 results, the cat, dog, and ferret, all have the qualities of being carnivores, are domesticated, and can have striking coat variations. BLAST is really useful because it can give us clues of how the protein functions across species, and therefore something to look out for if a mutation arises. So here we can see that we have a cat, and that is 100% alignment. And then we can see we have the dog, which is 85.5%. Then we have the ferret, 84.4%. Then we got the 2 types of pandas, and then a pig. And you can see all their similarity scores down here. So now we can take that protein sequence and find out a little bit more about it using the PDB, or the protein database. Unfortunately, this database is quite small and did not contain the LVRN protein for cats, dogs, ferrets, pandas, or pigs, so I had to use my BLAST results to search for similar organisms that were listed on the PDB site. The protein I ended up finding was isolated from Homo sapiens, or humans. We can see the 3D structure of the protein here, but we have to keep in mind that the sequence in humans is only 75% similar to the sequence in cats, so the actual aminopeptidase that the TACPEP gene encodes is probably going to look a little bit different. This protein is actually classified as a hydrolase, or an enzyme that catalyzes the hydrolysis, or the breaking up of, a certain substance. Back in the beginning of my presentation, I mentioned that the TACPEP gene is actually the first step in a 2-step catabolic process, and we can confirm that with this classification. To obtain the 3D structure of this protein, X-ray diffraction was used. This technique is performed by hitting the isolated protein with X-rays and then recording the resulting diffraction patterns of the rays. When the X-rays hit something that they cannot pass through, the wave diffracts, which results in an image that reveals the structure of the protein. If you're interested in looking at this protein more in depth, and you can actually do like a 3D rotating view type thing, it's very interesting, then you can use the PDB ID 4IRK and take a look for yourself. So here is that PDB ID if you're interested in looking at this in more 3D. This gene, though it may seem like it only has aesthetic value, can actually teach us quite a few things. I mentioned earlier that this mutation of the TACPEP gene, and therefore the protein, resulted in a receptor that rendered certain viruses unable to enter the cell. Therefore, this can give us a good idea of how mammals can defend themselves against viruses and increase their overall fitness. This also is a great example of epistasis, or how two genes, in this case the TACPEP and the EDN3 gene, work together to control a single phenotypic trait. Because we also know that the same gene exists and functions in cheetahs, they can also give us a good perspective on the phylogeny of cats and wild cats. So, the relevance of this class. A lot of content I've already presented relates back to our genetics course, and it doesn't stop there. Now, it's been understood that what causes the coat variances and that it's understood what causes coat variances, and that humans also contain the LVRN gene. Genetic engineering has actually taken place to further understand the role of this gene. Mice have been transformed, you can see in this picture right here, with the EDN3 gene, and had their mRNA levels monitored at different life stages and under different environmental stresses. We can also observe inheritance of traits with the TACPEP gene, as we notice passed on an autosomal recessive pattern. So we can see here that we have a heterozygous or homozygous dominant individual, with here being the heterozygous or the homozygous dominant, and this being the heterozygous. And then here we can see the homozygous recessive individual with the blotched coat pattern. And in the picture on the left, we can see how this gene expression is being monitored in light-skinned and dark-skinned kittens, and how dark-skinned kittens express the TACPEP gene and the EDN3 gene a lot more than light-skinned kittens. So here we see the amount of mRNA, and it's dark individuals and light individuals. Finally, we can take away from the universality of the genetic code amongst organisms, whether referring to transgenic mice, the same gene functioning in cat and cheetah, or the similarity of that sequence across species. This concludes my bioinformatics presentation. Thank you for your time and listening in.